In my research laboratory, we're trying to develop CAR T-cell therapies for AML, acute myeloid leukemia, because it's been proven to be very effective in acute lymphoblastic leukemia. However, it's a completely different disease entity, and there are many different challenges, one being that AML is a very heterogeneous disorder, both within a patient and between patients, and therefore to create a therapeutic which can meet the needs of most patients with relapsed refractory AML, you need to provide multi-antigenic targeting. And so this was work by my PhD student in our laboratory, who's generated an approach to try and target 3 different antigens that are relevant for AML. And so he's been able to generate a CAR construct that can sense the 3 antigens and effectively kill and activate CAR T cells after engaging with only one of those 3 antigens, for example. And this means that patients who have AML can be broadly targeted. You don't have to have a particular subtype of AML for this strategy to be relevant. And importantly, we're targeting CD33, which is present on most AML blasts, but we're also targeting CD123 and CLL1. These antigens are very important because they are selectively expressed on leukemia-initiating cells, which can be responsible for relapse in AML. And so as well as targeting the bulk population that express CD33, we felt it was important to have a broad coverage of antigens also targeting specifically the leukemia-initiating cells, hopefully with a view to eliminating those and preventing further relapse. All of these approaches, however, would be a bridge to transplant in this context, because these antigens are also expressed on normal healthy hematopoietic cells, particularly CD33 and CD123. And so unlike ALL, where we're trying to deliver therapeutics that replace transplantation, although that's an open question for the field, in AML, CAR T-cell targeting would be a bridge to transplant approach.